Good morning, everyone. Now, today, let's talk about the topic on lactose operon. Introductions. Operon is an operating unit which can be defined as a cluster of genes located together on the chromosome and transcribed together. It is a group of closely linked structure genes and associated control genes which regulate the metabolic activity. And these genes are called as polycystinic genes, and their regulation concept is also known as operant concept. In polycystinic genes, the regulatory sequence regulates number of genes simultaneously only due to the absence of spacer or non-coding sequence in between the consecutive genes. All these genes of an operon are coordinately controlled by a mechanism first described in 1961 by Francis Jacobs and Jacques Monod of the Pasteur Institute of Paris. So let's talk about what is lactose operon. The lactose operon is designated as lac operon, and the lac operon codes for enzymes that are involved in the catabolism of lactose, and it is a disaccharide which is made of glucose and galactose, and it is an inducible operon since the presence of lactose induces the operon to switch on. Here the structure of lac operon where we can see structural genes that are regulated by the operon. Here we can see the structures of lac operon where the gene Z, gene Y and gene A are the structural genes which where gene Z, as you can see, it encodes for beta galactosidase, and gene Y, that is lac Y, encodes for beta galactosidase permease, and whereas gene A that encodes for beta galactoside transacetylase, that is LAC-Z, and they eliminate the toxic thiogalactosidase also transported by LAC-Y into the cell, where gene Y, as you can see, it pumps the lactose into the cell, and gene Z, that is LAC-Z, that clips the lactose into glucose plus galactose. These are the functions of these enzymes that control the lac operon. And what is a promoter? The promoter is the binding site for RNA polymerase, the enzyme that performs transcriptions. Operator, it is a negative regulatory site bound by the lac repressor protein. The promoter overlaps with the promoter, and when the lac repressor is bound, RNA polymerase cannot bind to the promoter and start transcription. What is cap binding? It is a positive regulatory site that is bound by catabolite activator protein cap. When cap is bound to this site, it promotes transcription by helping RNA polymerase bind to the promoter. What is structural genes? It was discussed before that it has three genes, that is LAC-Z, LAC-Y, and LAC-A. What is regulation of lac operon? The lactose operon of E. coli encodes the enzyme beta galactosidase, which hydrolyzes lactose into galactose and glucose. The lac operon contains three cysteine on or are DNA fragments that encode a functional protein. The proteins which encode the cysteines may function alone or as subunits of larger enzymes or structural proteins. Here, as I have been told before, that Z gene encodes for beta galactosidase, and Y gene encodes a premius that facilitates the transport of lactose into bacterium, and A gene encodes a thiogalactoside transacetylase whose function is not known yet, and all these three genes are transcribed as a single polycystinic mRNA. Polycystinic RNA contains multi-genetic message, each with its own translation, initiation, termination, and signals. The activity of the promoter that controls the expression of the lac operon is regulated by two different proteins. One of the proteins prevents the RNA polymerase from the transcribing, that is negative control, and the other enhances the binding of RNA polymerase to the promoter, that is positive control. And this is the structure how they binds to the operator, how all this Z and Y and A transcribe into lactose. So now let's talk about a negative and positive control of lac operon. As we can see here in this picture, there is two diagram where we can see one is the negative regulation and the other one is the positive regulation. So now let's talk about the negative regulation. Uh, the negative regulation in absence of inducer, that is allolactose, the protein in this it binds or it inhibits the transcription of the lag operon is a tetramer with four identical subunits called lag operon, and the lag repressor is encoded by the lag gene located upstream of the lag operon and has its own promoter. Expression of the lag gene is not regulated and very low levels of lag repressor are continuously synthesized. Genes whose expression is not regulated are called constitutive genes, and this is in absence of lactose, and repressor is active and operon is off. And now in presence of inducer, that is allolactose, in absence of lactose, the lag repressor blocks the expression of the lag operon by binding to the operator. The operator consists of a specific nucleotide sequence that is recognized by the repressor, which binds very tightly, physically blocking the initiation of transcription. The lag repressor has a high affinity for lactose. When a small amount of lactose is present, the lag repressor binds it, causing dissociation from operator, thus freeing the operon for gene expression. Substrate that cause repressor to dissociate from their operators are called inducer, and the genes that are regulated by such repressor are called inducible genes. And this is the diagram, that is lactose present, repressor inactive, and operand is on. And now let's talk about the positive control of lag operon, that is positive gene regulation. Although lactose can induce the expression of lag operon, the level of expression is very low. The reason for this is that the lag operon is subject to catabolite repression or the reduced expression of genes brought on by growth in the presence of glucose. Glucose is very easily metabolized, so it, it is preferred fuel source over lactose. Hence, it makes sense to prevent expression of lag operon when glucose is present. The strength of a promoter is determined by its ability to bind RNA polymerase and to form an open complex. The promoter for the lag operon is weak, and consequently the lag operon is poorly transcribed upon induction. There is a binding site upstream from the promoter for a protein called a catabolite activator protein, that is cap. When the cap protein binds, it dissociates the DNA so that the RNA polymerase can bind more effectively. Thus, transcription of the lag operon is greatly enhanced. In order to bind a cap, must first bind cyclic AMP, that is CAM, a second messenger synthesized from ATP by the enzyme adenylate cyclase. In the presence of glucose, circulating CAM levels are very low, and consequently the initiation of transcription from the operand is very low. As glucose level decreases, the concentration of CAM increases, activating CAM, which in turn binds to the cap site, stimulating transcriptions. The CAM-CAP complex is called positive regulator. Here, as we can see in this diagram, in positive regulation diagram, where it has a cap binding site in it binds to the promoter. First, the CAM will bind to the inactive cap site, and it will get activated and it will bind to the promoter. Then the RNA polymerase that are bind with the promoter will transcribe and it inactivate the lag repressor. This is in presence of lactose. It it has high cam. It has high level of cam, and mRNA is synthesized. And the next in the second diagram, we can see the cap binding site binds to the promoter, where inactive cap can binds where RNA polymerase is less likely to bind to the promoter, and it has inactive lag repressor, which is lactose present, glucose present, where CAM level is very low, and it also synthesizes mRNA very less. So next, this is where E. coli that exhibit various abnormalities in lactose metabolism. Some mutants express the lack of operon genes constitutively whether or not lactose was present in the medium. Such mutants are called constitutive mutants, and this is R mutant lac gene, which uh, which has the phenotype constitute expression because the operator is never closed, or Operon lag gene where constitutive expression because the repressor protein cannot bind. P. No expression of the operand because RNA polymerase cannot bind. Lag Z. No glucose or galactose production from lactose. Lag Y minus no induction because lactose will not be taken into the cell. These are my references. Thank you.